In this lecture, let's start to discuss sources of genetic diversity for sexually reproducing species. And there are three major sources of diversity. The first is mutation. The mutation of genes gives rise to different alleles or new alleles in the genome. This is not something that's unique to sexual reproduction. However, mutation is a source of diversity for all organisms. The other two sources of diversity, which comes from meiosis and from fertilization, are unique to sexually reproducing species. So let's focus on those. There are three major sources of variation in meiosis. The first source of variation comes from recombination or crossing over in prophase 1. This is an image of two homologous chromosomes during synapsis. This special protein structure holds the two homologs together. By rearranging and creating different combinations of maternal and paternal DNA, or maternal and paternal alleles, this ensures that each of the daughter cells that result from meiosis will be genetically different. Keep in mind that while alleles are being exchanged, the position of the genes on the chromosomes does not change. A second source of variation is the independent assortment or random segregation of chromosomes in anaphase 1. This is best explained by using images shown here. There are two pairs of chromosomes here in this genome. The homologous chromosomes have paired up and they're at metaphase 1. The paternal chromosomes are shown in blue and the maternal chromosomes are shown in red. And you can see that by chance the two paternal chromosomes have landed on the same side of the metaphase plate. In this other cell, we have the same cell type with the two pairs of chromosomes, only in this case at metaphase, again by chance, the chromosomes have landed on the metaphase plate such that one paternal and one maternal chromosome faces each of the poles. These two different orientations of chromosomes at the metaphase plate will result in two different outcomes in the daughter cells. In the first case, one daughter cell receives both paternal chromosomes and the other both maternal chromosomes, and in the other case, the daughter cells receive a mix. In each case, the daughter cells are genetically different from each other because each has a different subset of the original maternal and paternal chromosomes. These differences in how the chromosomes land at the metaphase plate and are separated in anaphase is what is meant by independent assortment or random segregation. And it's called random or independent because the chromosome pairs themselves are behaving independently of each other and they're landing at the metaphase plate in a random orientation. So the independent assortment of homologous chromosomes in meiosis 1 results in a variety of different combinations of maternal and paternal alleles. And if we just start by ignoring crossing over for a minute, you can actually calculate the number of possible combinations of maternal and paternal chromosomes in the daughter cells as a result of independent assortment in anaphase 1. If n equals the haploid number of chromosomes, the number of possible combinations is equal to 2 to n. So here we have dogs whose haploid number is 39, and there are 2 to the 39th possible different combinations of maternal and paternal chromosomes that can happen as a result of independent assortment in anaphase 1. This means 550 billion different outcomes, and this does not even factor in the other sources of variation, such as crossing over. The third source of variation is similar to the random assortment in meiosis 1, only this pertains to meiosis 2. Here we're looking at the daughter cells that result from meiosis 1. And you can see that each one of these chromosomes has two sister chromatids, but the sister chromatids themselves are no longer identical because of crossing over, which happened in prophase 1. So here at the top we have a sister chromatid that's all maternal chromosome, and we have a second sister chromatid that's a mix of the two with a little bit of paternal DNA. This is also true for the smaller chromosome below it, and in the third chromosome we have a sister chromatid that is all paternal DNA with a second chromatid which is a mix. How these different chromatids land on the metaphase plate is also going to be a random event and can lead to different outcomes depending on how they land. And again, we can take this example up at the top here at metaphase 2. And I'll just get it in a larger format here. We can look at how the sister chromatids have landed at the metaphase plate. Looking at just the top 2 chromosomes, they have landed so that the sister chromatids that are all maternal DNA, this one here and this one here, are facing opposite poles. But they might just as easily have landed so that both of the chromatids that are all maternal DNA were facing the same pole. And if that were to have happened, the outcome for the daughter cells would be different. One daughter cell would get all maternal DNA for these 2 chromosomes, and the other daughter cell would get a mix of the 2. Similarly, if you look at the relationship between the bottom 2 chromosomes, again they have landed on the metaphase plate so that the chromatid that is all paternal DNA down here is facing the opposite pole to the chromatid in the other chromosome, which is all maternal DNA. And this means that one daughter cell that results will have the large chromosome, which is all paternal DNA, and the other daughter cell will have the smallest chromosome with all maternal DNA. But again, it could just as easily have happened that these 2 chromosomes would land on the metaphase plate such that the all paternal chromatid facing this pole was oriented to the same pole as the all maternal chromatid in the smaller chromosome. And in that case, the outcome would be different for the daughter cells, because one of the daughter cells would have both the large chromosome of all paternal DNA and the small chromosome of all maternal DNA. So different recombinant chromatids can land on different sides of the metaphase plate in metaphase 2, and this will lead to different genetic outcomes for the daughter cells. This gives rise to the potential for even more variation than we've already described. And finally, the act of fertilization itself can lead to genetic variation. If you think about the many, many billions of possible combinations of alleles and maternal and paternal mixes of DNA that can result from meiosis for the 3 reasons we just discussed, the final act of variation is which one of those billions and billions of possible combinations in the egg and which one of the billions and billions of possible combinations in the sperm actually find each other during fertilization and form the new individual. Without crossing over at all, there are more than 70 trillion possible diploid combinations of chromosomes in humans at fertilization. With crossing over, it is infinitely higher, and it becomes almost infinite. And this is why when you have siblings, unless they are identical twins, they're never going to be exactly the same. Although meiosis 1 is supposed to result in the separation of homologous chromosomes in anaphase 1, sometimes a mistake is made and this doesn't happen. And both homologous chromosomes can end up going to the same pole. When this occurs, this is known as non-disjunction. In other words, it's failure of chromosomes to segregate properly, and it can happen at either meiosis 1 or meiosis 2. In cases where non-disjunction occurs in meiosis 1, one of the daughter cells ends up with an extra chromosome, and the other daughter cell is missing a chromosome. Because both of these cells will in turn undergo meiosis 2, the result for the 4 daughter cells is that 2 of the daughter cells have an extra chromosome and 2 of the daughter cells are missing a chromosome. Whenever you have an extra chromosome or you're missing a chromosome, it's known as aneuploidy. When non-disjunction occurs in meiosis 1, all of the daughter cells are affected. When you have non-disjunction in meiosis 2, however, 2 of the daughter cells will be normal and 2 will be affected. So here is an example where the first division occurs normally. Both homologous chromosome pairs are separated properly. You form two normal daughter cells. Non-disjunction, however, happens in one of the daughter cells that result from meiosis I. The daughter cells that result from this division will be abnormal. One has an extra chromosome, and the other is missing a chromosome. The other cell, which was normal to begin with, undergoes a normal anaphase II, and it generates two normal gametes or sex cells, or daughter cells. So the outcome in meiosis II will be two normal daughter cells and two abnormal daughter cells. There is, not surprisingly, terminology associated with how you describe non-disjunction outcomes. Euploidy simply refers to the normal number of chromosomes. So if you're a euploid, you have the normal number of chromosomes for your species. Aneuploids refer to organisms that have an extra or a missing chromosome. And if you have an entire extra set of chromosomes, you're known as a polyploid. So organisms that have a whole extra set are triploids. Organisms that have two extra sets, or in other words, 4 sets of chromosomes, are tetraploids, etc. Often polyploids can result from failures during mitosis in the cells that will ultimately give rise to the germ cells. The germ cells being the cells that will undergo meiosis. Now, whether or not aneuploidy or polyploidy is going to be a problem for the offspring depends on the type of organism that you are. Plant cells tend to tolerate aneuploidy and polyploidy quite well, and if you go into a garden center, those flowers that are especially large and especially bright are often polyploids. But animals in general are much more sensitive to aneuploidy or polyploidy. In humans, for example, aneuploidy and polyploidy normally will result in the death of the embryo. There are some exceptions. One, of course, is Down syndrome, where there's an extra chromosome 21, and you can see this genome here from an individual with Down syndrome. These individuals do have some health challenges, but they normally do survive childhood and even into adulthood. Note, however, the size of chromosome 21. So this is one of the smallest chromosomes in the genome. The other source of aneuploidy which is tolerated in humans is related to the sex chromosomes. In the case of aneuploidy associated with sex chromosomes, no individual can survive without at least one X chromosome. However, individuals do survive with extra copies of sex chromosomes. Physical or cognitive development can be affected, but it will vary with the situation. In terms of aneuploidy where part or an entire chromosome is missing, those type of aneuploids are not viable in humans.